Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. First of all, I would like to express my grateful thanks to the organizing committee of the Third International Conference on Bioscience and Biotechnology, ICBB 2020, by Universitas Mataram University. And then this is my great pleasure to be here with you to share my research about the peculiar growth of Pseudomonas species LS3K with the addition of untreated tannery wastewater performed in Faculty of Animal Science, Universitas Gadjah, Mada, from me with the team. As the introduction, the tannery wastewater from the leather tanning industry will consist of organic and chemical pollutants, and it will need the complex step to treatment for the bioremediation. Bioremediation denotes as utilization of living organisms to degrade some hazard pollutant and contaminants, especially in the wastewater, to obtain the waste or the quality to improve the quality of the water. So one of the bioremediation agent which is proposed to be used in this research is Pseudomonas species LS3K, which is previously isolated in our laboratory and it has been registered in the NCBI also. So in this research, we want to know the characteristic of Pseudomonas species LS3K, especially growing in the presence of tannery wastewater. Well, as we can see here, this is the scanning electron microscope of strain LS3K, which is previously published in the Research Asian Journal of Microbiology, Biotechnology, and Environmental Science, to prove that actually this Pseudomonas species LS3K have the ability to nitrifying. Yeah, this is involved in nitrification process, which is change the compound, especially for the ammonium into the nitrate and the nitrate into the nitrate, which is usual take place in the treatment of the industrial wastewater. As we can see in this slide, and as we can understand that actually the tannery wastewater treatment will always consist of three step, which is very important, and it cannot be separate between one to another. The chemical process, the biological process, and the physical processes. We attempt that the LS3K will have some function in the bioremediation, especially to degrade the organic compound from the tannery wastewater. This is one of the schema which is previously we have studied about how to decompose the organic matter from the wastewater with using some microorganism. So the objective of this study, we want to characterize especially the growth of Pseudomonas species LS3K with the medium with the addition some untreated tannery wastewater. Especially this is we conduct this research in laboratory scale. As the material and method of this research, we have take the untreated tannery wastewater from tannery industry in Bantul City, and then for the purification, we conducted using the filter paper workman. And we continue also with the sterilized by the autoclave with the 121 degrees Celsius for about 15 minutes and 15 bar. The isolated Pseudomonas species LS3, which is using in this research, was again deposited and collected in the laboratory of Leader Waste and Byproduct Technology, Faculty of Animal Science, Universitas Gadjah Mada. In this research, we use agar medium, nutrient agar medium, and especially in the solid one, with the addition some various concentration of tannery wastewater. And then also we conduct the research using the liquid medium, which is contain some sterile fresh tannery with the different concentration of industrial or tannery wastewater. For the variables and analytical method, the growth of the cell was determined by observing the visual colony on the agar medium, and then also monitored by the optical density 600 using spectrometer, especially in the liquid medium. And we observe also what the cell viability after the cultivation in the liquid medium containing the tannery wastewater. And some description using figure graphical data was used in this study to try to conclude what the phenomena appear in the research in this research. Well, this is the growth characteristic strain LS3K on the solid medium especially. As we can see in this slide, in the day first day to the day fifth, and also the different concentration of the addition of tannery wastewater from 0, 25, 50, 75 till 100. And as we can see here, the growth was observed especially in the medium containing 0, 10, 25. Uh, Pseudomonas species LS3K also was able to grow. This is from the first day to the fifth day, especially for the zero. However, in the 25% of the addition of the tannery wastewater, it was appeared especially for the second day. And the growth of Pseudomonas species was not observed in the medium, which is contained of 50, 75, and also 100% from the first day till the fifth day. So this is important, and this is a good phenomenon for us that under the 25% of the addition of tannery wastewater, this strain still consists and can grow very well. And how about the characteristic of this terrain, especially in the liquid medium? Uh, as we can see here, this is a several graph. P1 means the addition of 25%. The addition of 25% is like this, and then the P0 mean the addition of 0% of wastewater. It will be like this, a good growth without any stressor from the uh, wastewater. And then also the P2, which is I can see here with the addition of 50% of the tannery wastewater without any addition of the bacteria. The growth is no different between from first day to 36 time of the observation. So as we can see and we can understand from the graph, and similar with the addition, the research performed in the liquid, in the solid medium, by the addition of 25% in the wastewater addition, it indicate that the it need, looks like the adaptive potential for utilizing nutrient from this strain. And then the resources of for growing, for growth and cell metabolic activity, which can be observed based on the increase of the absorbance value in the median. And the last data showing about the viability cell of the Pseudomonas species LS3K. This is for the first day, second day, and the third day. And as we can see in this table, that the viability test shown the addition of tannery with the P1 and P2, it means that the addition of 25%, and this is for the addition of 50%, it look like some inhibition in this growth of the strain. And the visualization of this colony formed after the cultivation of P1 and P2 were thinner. It look like not so thick and thinner, as we can see in this table also, between day first, day two, and day three. And the presence of tannery wastewater, it has some inhibition activity, uh, uh, well, according to the viability test of the Pseudomonas species LS3. And this is the last slide from me. Based on the result of the study, we can conclude that the Pseudomonas species LS3K especially had the potential actually to adapt it to live in the growth and the median that contain contaminated by up to 25% of fresh tannery wastewater. So this is a good result for us and good opportunity to share about the research regarding the growth behaviors of the strain Pseudomonas species LS3K with the addition of the tannery wastewater. Thank you so much for your kind attention.